Arpad Vass, Arpad Vass.
May I
You
proceed?
may proceed.
Good morning, Dr. Vass.
Hello.
Dr. Vass, you are a senior research scientist at Oak Ridge National Laboratory. Is that correct?
Yes,
And you live in Oak Ridge, Tennessee. Is that correct?
correct.
How long have you worked at the Oak Ridge National Laboratory?
Almost 20 years.
And what is your area of expertise in which you work at the Oak Ridge National Laboratory?
That's a complicated question.
Okay.
As a research scientist, we have to have general expertise in a variety of scientific disciplines.
All right.
My areas of expertise include anthropology, biology, chemistry, microbiology, clinical pathology, and endocytobiology.
Right. Tell us, if you will, your educational background that kind of led you up to working for Oak Ridge National Laboratory.
I have a bachelor's from Virginia Polytechnic Institute and State University in Blacksburg, Virginia. I have a medical technology degree from Fairfax Hospital, a master's in forensic science from Virginia Commonwealth University, and a PhD from the University of Tennessee in Knoxville in anthropology.
And your master's degree, you said, was in forensic science?
Yeah, administration of justice, I think, is with forensic science is the add-on.
Okay. Tell us, if you will, how did you end up getting your PhD at University of Tennessee?
While I was in Virginia, I was working as a medical technologist, and I was taking master's classes, going through my master's at the Medical College of Virginia and Virginia Commonwealth University. And I became very interested in forensic science at that point in time. I was offered a position at the University of Tennessee in Knoxville, and my wife and I moved down there. And I wanted to continue my education in forensics and get a PhD. And so at that point in time, I learned about Dr. William Bass. He had established in 1972 a anthropological research facility where he essentially studied the decompositional process in an outdoor setting. I became very interested in that and went and chatted with Dr. Bass and developed a PhD dissertation based on that conversation.
All right. Now, the anthropological research facility, that is the proper name for what in the media is referred to as the body farm. Is that correct?
Unfortunately, yes.
Okay. Not particularly, not one that the facility may like, but that's kind of what we call it.
Yes.
But we'll call it the correct name as far as we're concerned. Now, when was it that you began these discussions with Dr. Bass about pursuing a PhD in forensic anthropology?
Probably about a year after I arrived in Knoxville, give or take, maybe less.
What year was that?
I think 1988, 1999, 1988, 1989 timeframe.
Okay, very good. Now, when you were discussing pursuing your PhD, was it to study a specific aspect of forensic anthropology, or was this just a general study kind of thing?
No, it was a specific aspect of it. After chatting with Dr. Bass, I like challenges. And one of the challenges that he has always faced is determining how long someone has been dead, the post-mortem interval. Biochemically, prior to some of the work we have done, that has never been really studied in depth at all. And so my dissertation and thesis work was looking at the biochemical parameters associated with human decomposition to determine whether or not that could be used as a means of doing the post-mortem interval, or determining how long someone's been dead.
All right. Up to that point in the late 80s, was there a substantial sort of subfield within forensic anthropology of biochemistry, or was that fairly new?
It was fairly new.
Okay. So when you began your work, let's describe a little bit, if we can, for those that may not know, the anthropological research facility. It's a rather unique facility, or at least it certainly was back in 1988, 89. What's unique about the anthropological research facility? Explain that to us.
At that point in time, it was essentially the only facility in the world where you could study a whole body decompositional events. It's an outdoor facility. It's roughly a little less than 1.5 acres in size at this point in time. Again, it began in 1972. It was established. At this point in time, there have been roughly about 1,100 test subjects that have gone through that facility.
And by test subjects, we mean people who've donated their body to science, and it's then used for this research.
That is correct, yes.
And these are actual human bodies that are left outside under different... It's preliminary questions, Judge. Right. Are these actual human bodies?
Yes.
Are they left outside?
Yes.
Are they left outside under various conditions?
That is correct.
Some in cars?
There is a vehicle out there, and studies have been performed with individuals in the trunk of that car or interior in the car, yes.
Some buried?
Correct.
Some on surface?
Correct.
Now, your studies beginning in the early 80s, you were looking at something you called post-mortem interval. Is that correct?
Yes.
You said it was time since death.
Correct.
Now, what approach were you taking to solving that problem with your initial research?
The initial research was looking at the chemical breakdown of soft tissue. Soft tissue in the human body is made up of 4 components: proteins, lipids, carbohydrates, and nucleic acids. As the body decomposes and liquefies, that liquid pours out upon the matrix upon which the body resides. Could be soil, could be clothing, could be bedding, could be carpeting, wherever the body resides. We analyzed that fluid to determine what components in that fluid were reliable and reproducibly produced, which could indicate how long that individual had been dead.
Now, are there different stages of decomposition that you could describe to the jury?
Yes, I will try not to be too morbid. There are typically recognized 4 stages of decomposition. There's a fresh stage, a bloat stage, active decay, and then a dry stage. In the dry stage, remaining non-nutritive tissues, typically mummified, or it could be just skeletonized material.
So the first stage, the fresh stage, just kind of tell us what's going on with the body during the fresh stage of decomposition.
Okay. There are 2 processes involved in early human decomposition. One is called autolysis, from the Greek auto, self, lysis, digestion. So it's a self-digestion process. If someone dies suddenly, well, let me backtrack. There's about 100 trillion cells in the human body. If someone dies suddenly and traumatically, it doesn't matter if it's traumatic, but the cells of the body don't yet realize that the organism has dead or about to keel over and die, you see. So the cells continue to do what they normally do, and that's cellular metabolism. Now, and this begins, cellular metabolism continues on, but
the narrative, and this witness is not a biologist.
Go ahead, sir.
Does that mean I can continue? So the organism has died because of the traumatic death of the individual, but the cells of the body don't yet realize that the organism is dead, and they continue to do what they normally do, cell metabolism. One of the byproducts of cellular metabolism is carbon dioxide. Normally, the carbon dioxide is released or removed from the cells by the circulation and then the respiration. You exhale carbon dioxide. Because there's no more circulation, because there's no more respiration, the carbon dioxide really has no place to go. So it builds up inside the cells of the body because the cells are metabolizing. Carbon dioxide is acidic. Eukaryotic cells, the cells of mammalian bodies, don't like.
Both to the narrative, and this witness is not a chemist.
Your Honor, could we
Overruled. You may continue, Doctor.
go ahead?
Thank you. I forgot what it was.
You were talking about the being acidic.
Yes, carbon dioxide is acidic. It's building up inside the cells. Organelles, cells are made up of very complex structures. And these are called organelles. And this is the nucleus and things like Golgi apparatus and ribosomes and all these things.
Yes, sir.
If you want this taken down, and I'm quite sure that both sides are on the record, you're gonna need to slow down.
I'm very sorry, sir.
Okay, no problem.
Should I begin again? Okay. The complexity of cells depends on what's inside them, the anatomy of them, for instance. So inside cells of the body, there's a nucleus. There are other structures. One of those structures inside a cell, or an organelle inside a cell, is something called a lysosome. These are surrounded by membranes and contain enzymes. When the cell is normally functioning, the enzymes are used to digest food and get rid of cell waste products, things like that. Enzymes are proteins, and the definition of them is that they speed up chemical reactions. In the case of digestive enzymes, they're used to break things down. Now, a lysosome inside a cell is surrounded by a single membrane, unlike some of the other structures inside the cell. Because the cell is now becoming acidic, because the carbon dioxide is increasing, the first structures to be affected are single membraned organelles like a lysosome. So as that membrane ruptures, it releases enzymes. And these enzymes begin the process of digesting the cell from the inside out. This is autolysis, self-digestion. This is visually apparent by blisters forming on the surface of the skin and the organs, because now the cells are essentially liquefied inside because of the
Improper predicate.
Overruled.
You can go ahead, sir.
Okay, sorry. Because the interior of the cell is now liquefied, it's nutrient rich. And as these blisters rupture, you begin to see things like skin slippage, where the surface of the skin becomes loosened and sloughs off the body. This is one of the first visual signs that something is really amiss with the body, you see. Now, these nutrient-rich fluids give the body, as the blisters rupture, give the body a moisture-laden appearance, because the blisters rupture, the liquid is now on the surface of the body, and the body looks wet. This allows the second major process of early decomposition to begin, and that is called putrefaction. Putrefaction is where microbiological organisms, whether it's bacteria, viruses, fungi, whatever, now feed on those nutrient-rich fluids that are released from the autolytic process. And these organisms can come from inside the body, from on the skin, or from the environment. And those two processes alone begin the process of liquefying the body. And that's essentially what decomposition is. It's taking a very complex structure like what we are and breaking it down into smaller molecules, eventually causing a liquefaction and dissolution of the soft tissue. Sorry that was so lengthy.
Sure. No, lovely breakfast conversation, isn't it? You mentioned something called a bloat stage. You talked about autolysis, putrefaction, and bloat. What is the bloat stage?
The bloat stage is when microorganisms in the intestinal tract of humans again, they're metabolizing. They're really going to town because they're inundated with all these wonderful nutrients they never had access to before. So now these organisms are really going to town, and one of the byproducts of bacterial metabolism is the generation of gas. Okay, it could be hydrogen, it could be methane, it could be carbon dioxide, it could be ammonia, all these different gas. I'm sorry.
It's okay.
I'm sorry.
That's all right. So just start back from.
I love my subject.
I can tell. 1 second. Madam Court Reporter, where would you like me to start from?
Metabolism.
Overall,
And improper predicate.
overall,
You were talking about the gases. That's where I think she lost you.
Yeah, as the bacteria are now utilizing these nutrients that are now available, they produce, they actively make waste products as well, which are gases. And these include ammonia, hydrogen, methane, carbon dioxide, things of that nature. And when someone dies, occasionally the rectal region and the esophagus can become occluded. And
What does occluded mean?
Blocked, essentially. Now, when that situation happens, the gases that are produced from the bacterial metabolism have really no place to escape. And so it builds up in the abdominal region. And that causes the appearance of a bloated abdomen, if you will. Now, it doesn't necessarily have to happen only in the abdomen. Wherever bacteria are present and the gases are trapped by whatever mechanism, you will see a distension of the tissues, and that's called bloat. That will eventually find a way out as the body decomposes over time.
And the final stage you described was?
Well, there's active decay after the bloat stage.
Okay, active decay. Tell us about active decay.
Active decay is essentially the major, roughly 40 to 80% of the decompositional process is active decay. Active decay.
overall,
Active decay is when all the major aspect of the liquefaction of the soft tissue occurs. So all the organs are liquefied, all the nutritive tissue it becomes dissolved, and that leads to the final stage, which is the dry or skeletonized stage, where non-nutritive tissue, non-nutritive because if it has no nutrient value, the bacteria and insects really won't utilize it, and they'll ignore it. And so that remains. It can become dry and parchment-like, which is mummification of remaining tissue, or you can be just left with essentially a pile of skeletal elements or bones.
What is the one environmental, if there is one, what is the most important environmental factor in the time that process takes, whether it's faster or slower?
There are 4 processes that are important in the rate of decomposition. Temperature is the most important. The second is the presence of water, and the water can come from the environment, it can come from inside the body, humidity, things of that nature. The third is pH. pH is essentially a measure of whether something is acidic or basic. There's a pH scale which runs from essentially 0 to 14, 7 being neutral, or above 7, it's alkaline or basic. Below 7, it's acidic. Surface or aerobic decomposition, that's decomposition in an oxygen type of environment, like if you're just lying on the surface, typically is very alkaline, high pH. Burials or anaerobic type of decompositional events tend to be very acidic, like a burial situation, or if you're wrapped in something that you can't have a lot of oxygen exchange. And the fourth major component is the presence of oxygen. Again, the aerobic is much more rapid than anaerobic, which is lack of oxygen. So those 4 major parameters affect the rate and completeness of the decompositional process, temperature being the most important.
In that respect, would higher temperature accelerate decomposition?
That is correct.
Lower temperature might make it slower.
That is correct.
All right. During your study that you began with, looking at postmortem interval, what was it about that process that you found to be significant in determining postmortem interval? Let me back up for a minute. What were you looking at in your initial study in determining postmortem interval? In other words, the body itself or something the body was on? What were you looking at?
We were essentially looking at the liquefaction products. As the body liquefies, is there anything in that liquid that could be useful for time since death or postmortem interval determinations?
And how did you, just very briefly explain to us how it is that you did that study?
Well, essentially, we didn't really know what to look for. So we started screening for a variety of compounds. And what we essentially found was that fat and muscle breaks down into something called volatile fatty acids. Now, there's about 20 or so of these volatile fatty acids. Of those, what we found was there's only a few that are reproducible body to body and produce ratios that could be useful in determining how long someone's been dead. These fatty acids include compounds called butyric acid, valeric acid, and propionic acid.
Right. And did this study end in publication of your findings?
That is correct, it did.
And how long did you spend on the study before the publication of findings? And if it was more than once, just kind of take us through that history.
I'm sorry, Mr. Ashton, I didn't.
Did you just publish the findings of the study once, or was there a series of papers?
For the volatile fatty acid publication, there was just one publication on that. Now, included in that was another aspect of what we discovered during that particular study, and that was the analysis of inorganic components. I mean, the human body is composed of organic material and inorganic material, like calcium. That's inorganic, you see. And so we also found that a variety of inorganic components could be used to determine how long someone's been dead. Normally, the volatile fatty acids are used for earlier time since death estimates because there's a lot of soft tissue being produced. But as the soft tissue goes away, the inorganics become more important. And these inorganics include things like calcium, magnesium, sodium, potassium, compounds such as that.
Now, the first paper that was published on the volatile fatty acids, when was that published? If you can approximately remember.
I think it was 1992, I believe, in the Journal of Forensic Sciences.
Okay. And pardon me, the later follow-up papers involving the inorganics, when approximately were those published?
Again, that was one paper.
One paper.
The one paper included the volatile fatty acids and the inorganics. Now, we developed other methods of doing time since death. In my 20 years working at the decay facility, the first 10 years roughly was spent on developing a number of different types of methods because homicides never occur the same way twice. So we had to have a variety of tools in the toolbox available to us to be able to use different methods depending on the circumstances.
All right. Now, pardon me, you were doing the research at the anthropological research facility. Were you employed at Oak Ridge, but using the facility, or were you working at UT?
And during the volatile fatty acid paperwork, I was still an employee at the university. Okay. For later work, I was then an employee at the Oak Ridge National Lab.
Were you awarded your PhD based upon your work that you've described from UT?
Yes, the volatile fatty acid work, that is correct.
And when did you receive your PhD?
1991.
And now, when did you go to work for Oak Ridge?
92.
So just after your
Yes.
PhD. All right. So much of this later work was as you were employee of the Oak Ridge National Laboratory.
Cool.
Could you just tell the jury briefly what is the Oak Ridge National Laboratory?
Okay.
I know that's a big question, but give us a brief overview.
The Department of Energy runs a number of national laboratories across the country. Oak Ridge National Laboratory happens to be one of them. It is the largest research laboratory in the DOE complex. It has roughly 4,000 to 5,000 employees, a large number of guests, international guests and researchers come and perform research there. There are a number of key areas of research. Typically, these are very large, complicated projects which require multidisciplinary teams to investigate.
Overruled.
Go ahead.
These include things like climate change, looking at new types of biofuels, looking at the effect of neutrons and how neutrons can be used to investigate proteins or materials. Material science is a huge emphasis at the National Laboratory. Supercomputers, I think we have the second and fourth fastest computer in the world there. We have Homeland Security.
Doctor, let me interrupt for a second. I think you've just gone beyond the be brief part. So let me just stop you right there.
I'm sorry?
No, I said you've gone beyond the be brief part. So let me stop you right there. Does the laboratory have a broad range of scientific disciplines under the umbrella of the laboratory?
That is correct.
And the projects that the laboratory does are they beyond just energy-related items?
Yes, they are.
Now, your particular work with the laboratory, you talked about multidisciplinary projects. Have you been involved in projects that had nothing to do with forensic anthropology?
Well, again, as a research scientist, we have to pretty much have expertise in a variety of different fields. I may not have a PhD in those fields, but I have to know more than the average person walking on the street. So we have a number of different projects. For instance, we're looking at new types of plants that produce biofuels. Non-food crop plants, for instance. I mean, corn produces ethanol, but who wants to use a food crop to make a fuel, right? So we're looking at different types of plants. So I get involved with that because we don't know that these fuel products are actually produced in the plants and microbes in the plants. So one of my areas of expertise is microbiology. So a lot of these projects are multidisciplinary, lots of people involved with chemists, physicists, biologists, things of that nature.
All right. Did there come a point in time when you felt that you had done all you could do in the area of postmortem interval determination?
There was.
Overruled.
Go ahead.
Yes, there was. After the third model that we developed, and that was a model based on how tissue breaks down in very early decompositional stages. Protein is made up of building blocks of amino acids. And we were looking at the liberation of specific ratios of amino acids. And essentially that ended up being the cutting edge method worldwide of doing time since death, because we were on an hourly basis of determining how long someone had been dead. After that point, I pretty much said, biochemically, I've reached the limit of my knowledge. I don't really think I could do any more in that field. And so we became interested in other aspects of forensic anthropology.
When was it that you made that decision, approximately?
I guess early 2000, perhaps 2001.
Did another project garner your attention at that point?
Yes.
Go ahead.
As I said before, I like challenges. And up until that point, I thought that time since death was the most difficult question that a forensic anthropologist needed to answer. So that's why I like that area, because it's a difficult challenge. I was wrong.
And it's not responsive to the question.
Sustain.
Well, so why did you move into another area, and what is that area?
The other area of interest that I found interesting was the detection of clandestine graves. That is a very complicated process, especially if the graves are several years old. And it is always very problematic to locate a clandestine grave. And so we began looking at ways of investigating how to improve that type of technology to be able to identify where a body had been buried.
Up to that point, what was the predominant method for finding a clandestine grave?
Well, of course, the use of cadaver dogs, but primarily it was geophysical in nature. Ground penetrating radar, the use of magnetometers, hand probes. Hand probes are simple metal rods you push down into the ground looking for soil density changes. None of them were very effective. It depends on the environment you're working in. So we were looking or trying to determine if there was a more universal way that could be utilized in an overall toolbox of things you could use for different situations.
So how did you go about developing a method of exploring? Well, let me stop. So what method did you come to explore to find clandestine graves?
Because the most successful method at that time was cadaver locating dogs, we looked at odor evolution from decompositional events. And in order to study that, we utilized the anthropological research facility, the Body Farm, and we actually buried a number of individuals. Now, these graves were kind of special because we put in a piping system underneath the bodies, above the bodies, and at the surface of the soil. And these bodies were buried at various depths, down to 3.5 feet, I think was the deepest burial we had, and then there were some shallow ones. And we began monitoring which chemicals are being produced during a burial situation. And these chemicals are produced either at the body, that was the pipe underneath the body, or they could have been produced, then began migrating upward through the soil column. The soil column is the amount of soil over the body in a burial situation. And those would be then recorded in the piping system above the body. And then we looked at, finally, which of the compounds produced at the body made it all the way up through the soil column and escaped at the surface. Now, the importance of that is because those are the chemicals that a cadaver locating dog would have been able to smell. And also it allowed for the initial development or potential development of instrumentation that could detect those particular compounds.
Going backwards a little bit, since we're starting talking about odors, in your initial 10 years or more of working in forensic anthropology on your first project, did you have occasion to experience the odor of decomposing human remains at various stages?
That is correct.
Approximately how many times?
Well, over the years, and this may not be an exact answer to your question, but over the years in my 20 years roughly at the anthropology facility, we have followed approximately 50 individuals from start to finish. Start to finish means that they were fresh when they came in and they ended up skeletonization. So we were able to follow those 50 individuals. And we also looked at hundreds of individuals at a single time points, doing various procedures or experiments that we did over the last 20 years.
And in that period of time, have you found the odor of human decomposition to be a unique odor? just from a perspective of your smell.
Yes.
Is the literature on this subject, is there literature that comments on the fact of the uniqueness of the odor of human decomposition?
Are you referring to the work I've done?
Well, your work or other work of other people?
There are publications available that describe what the chemical components are that are liberated during human decomposition. We had 2 publications from the work we've done over the last 10 years, and the other researchers, Dr. Milt Stathopoulos in Greece has done this type of work with whole bodies. A number of researchers have done work with just individual tissues. There are a number of researchers that do work with pigs and do pig and human comparisons, things of that nature.
Well, that's a good point. Have you had the experience of also doing work with animal decomposition and the odor of that?
Yes, at the decay facility, primarily what animals other than humans that they have there include pigs. A lot of people in foreign countries don't have access to a decay facility, and they're only allowed to work with animal remains. And the animal of choice is usually a pig. So when they do human-pig comparison studies, those are the ones they utilize. I hate to admit it, but normally when we go and look at other animals, they're typically roadkills. We do live in Tennessee. So, you know, we have deer, we have dogs, we have cats, things of that nature that we have sampled over the years in terms of looking at how they produce odors.
Have you found the odor of human decomposition to be distinguishable, just from your nose perspective, from all those other things?
I'm sorry, Dr.
No, it's okay.
Ashton.
Have you found the odor of human decomposition to be distinguishable from the odor of the animal decomposition that you have experienced?
I think, yes. Animals tend to...
For speculation.
Overruled.
Go ahead.
Animals tend to have a more muskier scent. Domesticated animals like a pig have a much sweeter scent than human remains.
Right. Now, you were describing to us your study using the buried bodies. Over how long a period of time did you collect data from that study?
It began in 2002, and I believe it ended early 2007.
And did you publish an initial paper of your findings in 2004?
Yes.
And was that published? Where was that published?
The Journal of Forensic Sciences.
And is that a peer-reviewed, respected publication in the area of forensic science?
Correct.
Now, did you continue your your research after 2004?
2000.
After the 2004 paper, did the research continue?
I'm sorry, yes. The 2004 paper was when the establishment of the graves, the beginning of a assimilation of the chemicals that we identified, and an evaluation of the environmental parameters that affected the evolution of those compounds. And the environmental parameters include temperature, rainfall, barometric pressure, things of that nature. Now, it is interesting, and if I may elaborate just briefly,
Sure.
Barometric pressure, for instance. I mean, the ground is a living, breathing entity, if you will. So if a high pressure moves in, it pushes chemicals down into the ground. So you can't detect them at the surface. It doesn't mean that they're not being generated by the body. It just means that the high pressure is pushing them down.
Narrative, improper predicate, and non-responsive.
Keep going.
They're pushing them down into the ground. If a low pressure comes in, it seems to suck the chemicals up out of the ground. So it's a very complicated process. Looking at rainfall, for instance. I mean, in that initial first 1 or 2 year study, I can't remember exactly how many chemicals we saw, maybe 200-300 chemicals perhaps. These chemicals, some are water soluble, some are not water soluble. When it rains, sometimes they float above the water, sometimes they're pushed down to the ground. So it's a very complicated process of how the environment interacts with the chemicals as they're liberated during a decompositional process. In my second paper, what we did is at that point, we had been looking at bodies for roughly about 4 years in the ground, and they were by that time skeletonized. So,
To the narrative and non-responsive to any questions.
Okay. Approach the bench for a minute. I'm with the court reporter.
Your Honor, that court reporter could remind us where we were. I don't.
The last full question was, after the 2004 paper, did the research continue?
We'll pick it up there as soon as she's ready.
Just a second. You may proceed.
Okay, so the last question was, did your research continue after the 2004 paper was published?
yes.
All right. And did it basically continue in the same form as far as the buried remains goes?
Yes and no. And obviously, the study with the buried individuals continued, but at that point, we were starting to also look at individuals that were decomposing on the surface. We also included skeletal elements or bones in that study, and at the very end of that, we included another fifth individual that had been recently buried, looking at a different type of technology.
Okay, I think I missed the last couple words what you just said.
A different type of technology. We were using, instead of triple sorbent traps, we were using SPME fibers at that point.
Okay. Now, the
and SPME fibers, SPME, SPME.
and that's an acronym for
It's an
actual name. We call it SPME.
solid phase micro extraction fiber. I don't know if you want to get into that, but
That's fine. All right. Now, your work with the above ground bodies. Tell us, where did that take place? Was that at the anthropological research? It's hard to say. Anthropological research facility also?
you're allowed to say body form.
I wouldn't dare.
Yes. Yes. So for that study, we looked at about 10 surface individuals. Some were just lying on the surface. Some were loosely covered with plastic tarps, and some were actually encased in body bags. So those are the surface individuals we included in that study.
Obviously, since it's above ground, you didn't use the tubing system. How did you obtain information or samples from those bodies?
Even in the buried individuals, in the piping system, they were little ports on the ends of each pipe. I'm not sure if we're going to show a picture of that or not, but nevertheless. And what we did is we collected the air containing the odor on something called a triple sorbent trap, or TST. Now, a TST is about 2.5, maybe 3-inch long metal piece of material filled with 3 types of essentially activated carbon. Now, activated carbon, if you have freshwater aquariums, is one of the things that you use to filter out ammonia out of the water, the fish urine and things like that. But nevertheless, so this little piece of metal has 3 types of this activated carbon in it. And as the air passes through the triple sorbent trap, it essentially purifies the air, removing the odor components and capturing them on the activated carbon. And then you take that back to the laboratory and analyze it. The same procedure was used for the surface individuals, but instead of attaching it to the piping system, we either placed the tube inside a body bag, underneath the tarp, or dangled it close to the individuals on the surface. that were not covered.
And how do you get air to go through the tube?
Oh, I'm sorry, you use a little pump, a little air pump
Right.
that draws air from the environment through the trap and then out the pump.
And then how do you analyze the compounds that have been captured by the tube?
The tube is then removed from whatever we're sampling at the time. The ends are sealed with little nuts, and you tighten those with a wrench. You bring that back to the laboratory, and you go through a process of thermal desorption. What that essentially means is you heat up that metal tube. And you heat it up, and you have a flow of gas going through it, an inert gas like helium. And that, as the tube heats up, the activated carbon material releases the odor compounds. They're carried out of the tube by the gas, and they go into a gas chromatograph. Now, we also use the process of cryofocusing. What that means is as they leave the little triple sorbent tube and go into the analysis instrument, they go through a teeny little metal loop. And that little metal loop is essentially cryogenically frozen by liquid nitrogen. Now, liquid nitrogen freeze, the temperature is at 77 Kelvin, which is minus 321 Fahrenheit. So it's bloody cold. And what that does is essentially condenses and freezes all the odor molecules into a small little pellet. And then so it's a concentration type of a technique. And as you remove the liquid nitrogen and allow the tubing to heat up and warm up, then all these compounds now become volatile again and go through the instrument and allow for the detection and identification of the compounds.
Does that permit you to detect even very minute amounts of compounds?
That is the point of cryofocusing. It's to concentrate the sample. So the answer is yes.
Right. And these processes, triple sorbent traps and the cryofocusing, were these used in the buried remains and the above ground remains both?
That is correct.
For how long a period of time did you study the above ground remains?
2 to 3 years, perhaps.
2 or 3 years?
Give or take. I mean, it was like from 2004 to maybe 2006. Maybe it was about 2 years.
In the initial stages of decomposition of both the buried and unburied remains, how frequently would you take samples?
That's a good point. I believe initially we started weekly, switched to bi-weekly. As things became skeletonized, we went to a monthly type of a schedule because things were changing, but in a burial situation, it doesn't change very rapidly. The surface individuals, we probably did more frequently, probably weekly, bi-weekly.
All right. Now, at some point, did you update the findings in your 2004 paper with new information?
That is correct. That information was included in the 2008 paper.
Now, did there come a time in 2008, well, before I do that, Your Honor, at this time I would submit Dr. Vass as an expert in the area of forensic anthropology, specifically the biochemistry of human decomposition.
We would object and renew all previous motions and objections.
Okay. The doctor will be accepted as an expert witness in that field. You may continue.
Thank you. Now, sir, did there come a point in 2008 where you were contacted by law enforcement about possibly utilizing the expertise you've talked about in examining some materials from a criminal case?
Yes.
Who first contacted you, if you recall?
I believe it was Detective Yuri Melich.
Yuri Melich. Did you understand him to be from the Orange County Sheriff's Office?
Correct.
Did you have don't tell me what he may have said, but did you have a discussion with him about what they had and how you might be able to help?
I believe so. I don't remember the exact conversation.
All right. At some point, did they begin to send you some items of evidence to examine?
Yes.
All right. May I approach the witness, Your Honor?
You may.
Do you recall recall, Doctor, what the first item you were sent was? Do you recall the order in which you were sent them?
I believe it was that a metal evidence can.
All right. Let me show you what is marked as State's Exhibit I-O identification, and ask you, do you recognize the outside of the box?
No, it looks like a box. I mean, I know we call that's what I just don't.
Box?
Yes, I believe I did.
If I may. All right. I'm going to open it and have you look at the contents. Let me throw this tape.
Yes, you may.
Oh, sorry. Yeah. Okay, let me show you the contents of State's Exhibit I-O for identification. If you could just look at the can, but don't show it to the jury yet. It's not in. Just take it out, look at it, but keep it down below so the jury can see it.
Okay.
You recognize it?
Yes.
Is that the metal can that you were sent that you just referenced?
It appears to be yes.
Do you recognize it from? What do you recognize it from?
Well, the label and the can, and it had the evidence tape on it.
Okay. This time, Your Honor, we United States Exhibit I-O for identification.
What says the defense?
I'm going to check at this time. Robert J. President.
Let me see that for just a second.
Dr. Vass, when you received this item, was it in a sealed edition?
Yeah.
Your Honor, at this time, the state moved into evidence. State I-O identification.
We just renew all previous motions and objections.
It will be received in evidence as states numbered. Mr. Ashton, at 15 past the hour, we're going to recess for 15 minutes. Got about 4 more minutes.
At some point, did you receive at some point, did you receive an actual plastic bag with an air sample taken from the trunk?
I believe that is correct, along with the SPME fibers from Dr. Sigmund, I believe.
All right. The plastic bag with an air sample, did you actually utilize that in your testing?
I personally did not. I think Dr. Marcus Wise analyzed that. We were not really sure about.
Witness testifying about another individual's work.
Overruled.
Go ahead.
We were not sure exactly how that particular sample was collected. It wasn't by a normal procedure. Again, I don't recall what the results of that particular analysis, even if it was run, were.
If it was run, it wasn't used in your coming to opinion in this case.
That is correct.
Did you, at some point, ask the Orange County Sheriff's Office to take air samples from various the car, various parts of the car or the garage, or various things involved in the case?
That is correct, I did.
Did you send them some equipment in order to accomplish that?
Yes, I did.
And what did you send them?
I sent them an air pump used to collect air samples through triple sorbent traps, and we sent them triple sorbent traps.
Now, the air pump and the triple sorbent traps, are those the same devices that you previously described using in your studies of both buried and unburied remains that you've talked about for the last hour or so.
The triple sorbent traps, yes. This particular pump was a portable pump rather than a plug-in electric one that we normally use at the decay facility. It was just a more portable one because I didn't know whether they had access to electrical power where the car was.
So the pump is just the device that pulls the air through the trap.
It's like a small little. It's a box, maybe the size of a shoebox, that has two like aquarium air pumps in it.
All right. But as far as the actual device, the triple sorbent trap, it was the same one you used, same kind of device you used in your studies.
That's exactly correct.
Now, did you receive back from the sheriff's office the tubes from various locations relevant to this case?
The triple sorbent
triple
traps?
sorbent trap.
That is correct, yes.
Let me show you a number of items. We all. Oh, wrong glasses.
You too.
Make your point. Let me show you state's exhibits, a bunch of state's exhibits here. What I'm going to ask you, I'm going to read them out in the record. I'll ask you if you recognize these as the packaging that these triple sorbents were sent back to you in. NF and G and H and I and J and K and L and M. Again, are those the packaging that you received the triple sorbent traps from the sheriff's office in that you later tested?
That is correct. My initials are all on each individual packet.
This time, Your Honor, I would move into evidence the number of exhibits. Look at it.
You may.
And F and G and H, I and J and K and L and N and M. They would move all these items in depth.
What says the defense?
No objections, Your Honor.
They will be received in evidence as
Please, and F will become stage 115, and G will become stage 116, and H will become stage 117, and I will become stage 118, and J will become stage 119, and K will become stage 120, and L will become stage 121, and M will become stage 122.
Thank you. I think it's be a good time for a break, Judge, if you want.
yes. Okay, members of the jury, we're going to take a 15-minute recess at this time. I'm going to ask you not to discuss this case among yourself, nor with anyone else. Please remember all of my previous admonitions. We would take a recess to 20 minutes to the hour of 11. Okay, stay recognized, brothers of jury.
Yes, sir.
And does the defense?
Yes, sir, we do.
Okay, you may proceed.
Thank you. Doctor, let's get back into the analysis. Now, in the actual analysis of the various items that you were sent, were you assisted by other members of the Oak Ridge National Laboratory who work in other disciplines?
That is correct.
And who else assisted you?
Dr. Marcus Wise, he's an analytical chemist, and Dr. Madhavi Martin, who's a physicist.
Right. Now, let's begin with Dr. Wise. What was Dr. Wise's role in assisting you in the analysis of the evidence in this case?
Well, essentially, the laboratory where the instrumentation used to analyze these samples was located was his laboratory. And he's in charge of making sure the instrument's working properly, and he's an analytical chemist, and he's trained in interpretation of chromatograms and things of that nature.
This discussing another witness's actions.
Sustain.
Your Honor, if I may, as an expert, I would submit that the witness is allowed to utilize hearsay.
He can utilize it, but I'm not going to permit him to testify about somebody else's qualifications. He can testify.
All right, thank you, sir.
Okay.
Did he assist you in performing, what instrument was used to analyze the odors from the items in evidence?
A gas chromatograph mass spectrometer.
And there's a device that is that the device you're talking about that was in his laboratory?
I'm sorry, I couldn't hear you.
Is that device the device you were speaking about that was in his laboratory?
That is correct.
Now, when states exhibit in evidence 114, which is the can containing the carpet sample, when that was arrived in your lab, what was the first analysis that was done on that item?
The first analysis was an injection of about 0.8 of a mil of headspace directly from the air inside the can. The can lid was...
With this witness discussing about tests run by other experts, we would cite Melendez-Diaz versus Massachusetts. We'll have further argument.
Overruled.
Go ahead.
I'm sorry, I forgot where I was.
You were talking about how the initial test was done.
The can lid was just cracked. A syringe was inserted inside that little crack, and 0.8 of a mil was removed with the syringe, and that was then injected into the gas chromatograph, GCMS. That's the acronym for gas chromatograph.
And who did that?
Dr. Wise.
And in that particular initial stage, was any cryotrapping or concentration used in that initial step?
No.
And did he share with you the results of that initial evaluation of the air sample?
Yes, we looked at the chromatogram.
Melendez-Diaz versus Massachusetts.
Could we have a cite, Your Honor, on the case? Counsel is going to rely on it.
Is that the case you cited the last time?
Yes.
Give you the cite in a minute.
Citation is 129, Supreme Court, 2527.
Thank you. May I proceed?
You may.
Thank you. So what were the results of that initial evaluation of the unconcentrated air sample? Before I ask you that, you said something about headspace. I don't know the jury would understand. What do you mean by headspace?
The headspace is the volume of air contained within the can. I mean, there are items in the can, in this particular case, a piece of carpeting or spare trunk liner, and that occupied a very small amount of the can, and the rest of the can was filled with air. And the headspace is essentially what was the air above that sample in the can.
And what were the results of that initial evaluation?
This is outside of the scope of this witness's both knowledge and expertise, and we would request a sidebar to discuss the Melendez-Diaz.
Okay. Okay, you may continue.
Dr. Vass, did you?
The previous testimony of this witness relating to the areas discussed at sidebar.
No.
Dr. Vass, did you go over the chromatogram of the initial evaluation of the state's exhibit? We've been talking about the can.
With Dr. Wise, yes.
With Dr. Wise, okay. Did those reflect the presence of any chemical compounds?
Yes.
What chemical compounds did that initial evaluation show?
Well, it showed one particularly large peak and a large number of very, very small peaks.
Okay. What was the large peak?
The large peak was identified as chloroform.
Chloroform. Is chloroform a substance that you have Do you need me to stop for a minute, Judge?
No.
Oh, I'm sorry.
Yeah.
Okay. Is chloroform a compound that you have had dealings with in your research on anaerobic decomposition?
Yes, chloroform is a decompositional event product that we've identified in human decomposition.
So, having done that, what was the next step that you recommended or that you performed?
Okay, the reason we progressed is because the chloroform was shockingly high, unusually high.
As to this witness's testifying outside of his area of expertise.
Overruled.
Okay.
Because of that and the fact we saw a variety of very small peaks that were just if you have very, very small peaks, that they can be difficult to identify. So the next step we decided to do was to try and concentrate the air sample to try and get a better representation of what may be coming off that the evidence.
And where did you decide to use the same technique you had used in your other studies, the cryotrapping?
Yes.
And were you present for the next round of testing?
Yes.
So you were with Dr. Weiss in the room as it was being done?
Yes. I mean, during the analysis, yes.
Yes. Okay. So tell us how that analysis went.
Well, the way we did that is we actually removed the piece of carpet from the metal can. We did that because we weren't sure whether or not the air in the can was representative of what was...
Melendez-Diaz violation.
Overruled.
Go ahead.
I forgot what I was saying. Okay, we removed the carpet from the metal can because we weren't sure whether or not the air in the can represented what was coming off the carpet. So we took the carpet sample out of the metal can and placed it in a plastic bag called a toddler bag. That toddler bag is a polyvinyl fluoride type of plastic. It is specifically designed or manufactured or sold as a product to contain air samples for such testing. So we took that carpet sample and placed it inside the bag and incubated it for 2 days at 35 Celsius, essentially body temperature.
Let me stop you for just 1 second, ask a couple of questions. Is the toddler bag used because it is non-reactive? In other words, that it won't change the air sample composition. Is that why you use the toddler bag?
That is correct.
Okay. What is the purpose of incubating the carpet sample, as you said you did?
Well, one, because the carpet sample came from the trunk of a car in Florida. I mean, I don't live in Florida, we live in Tennessee, but I imagine in the summertime it's quite warm. And so we anticipated that the trunk temperature would be quite warm. We didn't know exactly what it was. We estimated it being in the high 90s, close to 100. Plus the fact that as you incubate this under temperature, it speeds up the evolution of these compounds or the volatility of these compounds. So we selected 2 days at 35 Celsius as essentially our procedure for doing this type of analysis.
Was your purpose to actually get the substance in the carpet to be released into the air?
Yes, we wanted to determine absolutely what compounds were being liberated from the carpet sample specifically, without any interference from what may have been in the air when the carpet material was collected or something of that nature.
And having done that, how do you then take an air sample from the bag, and then what came after that?
Yes, from that bag, which contains a liter of air, it's 1,000 milliliters, we extracted 10 milliliters and injected that through the cryotrap concentration procedure and did a GCMS analysis of that.
And did that additional procedure allow you to identify additional compounds that perhaps weren't identifiable in the first run that wasn't concentrated?
Yes.
And what was this, were you present for all of that process?
I believe for almost every one of those previous post ones, yes.
Yes.
I believe so.
All right. And what was the result of,
This witness has testified to almost everyone.
I believe he said he wasn't there.
Overruled.
All right. What were the results of your concentrated analysis of the carpet sample?
We were able to identify 51 individual chemical components in the 10 mils extracted out of the toddler bag.
Was one of those chloroform?
Yes.
Did in looking at a GCMS, what is the output of a GCMS called?
A chromatogram.
Chromatogram. Did the chromatogram show you something, show a particular compound as being in much larger abundance than others?
Yes, I mean, large peaks, tall peaks indicate a much more concentrated amount of material in a very small peak would.
What was the largest peak that you found in this evaluation of the carpet sample?
The largest peak in a chromatogram is called the base peak. The base peak was chloroform.
Did the amount of chloroform that you found in the chromatogram surprise you?
We were shocked.
Why?
We have never seen chloroform in that level in environmental samples before. At least I've never have in 20 years of shooting these types of samples.
Now, you did say that chloroform can be a byproduct of decomposition, correct?
Yes. In human decomposition, we have seen chloroform in a concentration level of parts per trillion. So very small amounts.
And this particular amount, let me stop. Did you attempt at some later point to actually specifically quantify the amount of chloroform that was in the air sample from the carpet?
It was kind of a byproduct. We shot a standard of chloroform. The standard is just a known concentration of an analyte, in this particular case, chloroform. The main reason to inject that was to verify 100% that the peak was indeed chloroform. But because the concentration of that standard was also known.
This witness is testifying outside of his expertise.
Overruled.
Go ahead.
Because the concentration of that standard is known, we were able to make a rough approximation. We did not quantify, get the exact quantity of chloroform present.
And based upon that, what is the rough approximation of the amount of chloroform in the carpet
Objection,
sample?
judge calls for speculation.
Overruled.
The concentration of chloroform in that sample was in the parts per million range.
And I believe you said that what you have seen in decomposition was parts per trillion.
Trillion, correct.
So it's million, billion, trillion.
Correct.
I have that right. All right. Did you attempt to do any more precise quantification of the amount of chloroform to determine exactly how much it was?
No, we did not. That would have required the purchase of a more concentrated standard. And after discussions, we realized or considered the fact that the amount of chloroform only represented a minimum amount that might have been present on the carpet anyway. So further quantification really wouldn't have been more helpful to us.
And when you say a minimum amount, pardon me, when you say a minimum amount, what do you mean by that?
Chloroform has a high rate of evaporation, very similar to gasoline. So chloroform, let's say, was formed or appeared in the trunk of the car at some point in time.
This is testifying against outside of his area of expertise.
Overruled.
So chloroform appeared in the trunk of the car at some point in time in the past. So over time, it's evaporating. And so maybe, here the car is at the tow yard, and here the car, it's evaporating, now it's in the Anthony home again. And now it's in the garage, forensic garage. And then all of a sudden here, a sample was collected, and at that point in time, after all this evaporation had taken place, we were in the part per million range, but we had no idea how much was present in the past. So we didn't feel we could make even a better approximation of that.
Okay. So, but the minimum is in the parts per million range.
Million range.
Okay.
Yes.
Now, did you also obtain and test a piece of carpet from a similar vehicle for purposes of determining whether chloroform was just naturally present for some reason?
Yes, we did.
All right. And did you run a similar test on that?
Correct.
Let me show you what's been marked as State Exhibit NP for identification and ask you if this is a document containing the chromatograms from both the Anthony, the evidence carpet and the control carpet to kind of demonstrate the differences.
That is good.
Would it be helpful for you in demonstrating the differences you saw in using this diagram?
Yes, it would.
This time, Your Honor, I move into evidence States Exhibit NP.
What says the defense?
We'd object. This is, the witness has not testified as to where he obtained this, who conducted the tests, and anything relating to its reliability.
Diagrams from the trunk sample carpet and the control sample carpet.
Doctor, who conducted that test?
Both Dr. Wise and myself conducted the test.
And is that the type of stuff that you reasonably rely upon in formulating your opinion?
I'm sorry, I don't understand the question.
Is that the data that you reasonably rely upon in formulating your opinion in this case?
Yes.
Okay. Anything else, folks? Okay. It will be admitted in evidence.
Published to the jury?
You may. Over objections by the defense.
All right, Doctor, and if we could, Mr. Perez, focus in on the top graph first, and we'll let the doctor discuss that one and see how this works. Okay, now, what we're looking at here, is this the chromatogram that is the actual output of the GCMS?
That is correct.
Now, can you, and again, I believe if you just want, you can touch the screen and make a little circle if you want. Show us which one of these peaks represents the chloroform.
I don't know if it's going to work, but this peak here, oops, how do I fix that?
That's all right. It's tricky.
The base peak, the tallest peak at roughly 16 minutes.
That's okay. We got it.
Yeah.
You can stop.
Sorry.
And now there are some numbers off to the left.
Yes.
That say abundance. What do those numbers represent?
Those are rough approximations of how much material is present.
Okay. The actual numbers are, what is the top number there?
The top number appears to be 1.6 times 10 to the 7th, so it'd be 16 million in terms of what we consider a rough estimation of how tall that peak is.
16 million counts of whatever.
Counts of whatever, yeah.
Of whatever it is. Okay. So that peak is appears to be somewhere above the 16 million-ish range.
Correct.
Okay. Now, and this is the sample from the carpet from the vehicle involved in this case.
That is correct.
Now, does this also show the other peaks that we'll talk about later that are significant in your determination?
Well, there are a number of peaks that, without going back and being able to identify, but this region here, that represents a variety of compounds associated with gasoline. Gasoline is a mixture of dozens and dozens of components: benzenes, xylenes, toluenes, pentanes. All these different compounds make up gasoline. And in a chromatogram like this, each one of those compounds can be separated. But that area or region represents the compounds associated with gasoline.
Now, are there some overlapping compounds between what's in gasoline and what your studies have shown are in the odor of human decomposition?
That is correct.
But you would you be surprised to see some gasoline components in a car?
No, in fact, my understanding, at one point in time, there was an actual gas can in the car.
All right. But this reflects the relative abundance of the components of gasoline odor as opposed to decomp odor and or chloroform.
That is correct.
Could we now look at the second chart? Now, this reflects a chromatogram from a vehicle sample that was obtained at your direction, correct?
Correct.
And where did you direct the person to get a sample from?
This was from a junkyard in Knoxville, Tennessee.
Okay. And did you instruct the individual to find a car of a similar make, year, model, et cetera?
That's correct.
All right. Now, this chromatogram, does it also show a peak for chloroform?
It does.
Could you just, let me hold on. Let me clear the screen.
I got a lot of blue little things all over mine.
I got rid of them. You can start over. Show us the chloroform peak on this chromatogram.
Again, the retention time, that is when chloroform comes off the instrument. It always comes off at a specific time. That's called the retention time. And that's 16 minutes, so that would be, whoops. Like, can I waste that and start over? I'm not good at this.
Hold on. Okay, try again.
No.
You know what? Why don't you let me do it for you?
Thank
Maybe
you.
I can. Hold on.
I don't have it needs a pencil.
This one, right?
Yeah, actually, right there.
Okay.
where the pen is now.
Okay, that's fine.
And
That's
if you
fine.
can get rid of it, you can actually see it now.
Okay, well, let's clear it so we can. There we go. All right, that's fine. Now, in this chromatogram also shows the presence of chloroform in this control carpet, uninvolved piece of carpet. Correct?
That is, well, the carpet was from two different vehicles. So we put the carpet from two different vehicles, control samples from the junkyard in the toddler bag, incubated for 2 days, 35 degrees Celsius, pulled out 10 mils, did the exact same procedure as you did with the carpet sample from the Florida vehicle.
Just for comparison, what level of chloroform was found in the control carpet according to this chromatogram?
Well, the chloroform is actually a percentage of that particular peak. But if you go over here and look at the scale, you can see that the peak height of that particular little lump at 16 is probably no more than 5,000 at the most in terms of height abundance.
All right. And that is as compared to
16 million that we
16 million.
saw in the trunk.
Right.
Now, I do have to say that, if I may just elaborate just a
Please
moment.
do.
The peak height is just one thing you need to look at. Normally, when you quantitate something, you look at the area under the peak. It's not just the height, but the entire area under there. And, you know, we used computer software to obtain the area under that particular peak or chromatogram. For the Florida carpet sample, that ended up, the area under there, even though it was 16 million tall, the area was about almost 400 million. So it was like 384 something million. The area under this particular peak in the control sample from two different cars was 10,000. And now we do see chloroform in environmental samples, from soils, from vegetation, but it's typically,
Overall.
Go ahead.
but that chloroform level that is represented by this control sample is very typical of what we see in environmental samples. You know, we're talking about very low amounts. We essentially call this trace. Trace means it's kind of at the detection limits of the instrument. And you can see a little hump, but it's not very big. That's what we consider trace.
All right. Now, think we've discussed that. We can unpublish that. Now, having completed the evaluation you've just discussed, did you have the carpet submitted to an additional kind of testing to determine whether the presence of inorganic compounds?
That is correct. We use the technique called LIBS, which is laser-induced breakdown spectroscopy. Now, both this Tedlar bag experiment and the LIBS analysis were intentionally done because they're both non-destructive techniques. You don't have to destroy the carpet in order to get these results. And we did that intentionally in case the defense or whoever wanted to verify these particular results.
How does LIBS work? Just give us a general
Okay.
idea of that.
If I go into it, let me know, I'll slow down.
Sure.
LIBS essentially uses
The witness is not a physicist and is now testifying in the area of physics.
Overruled.
On a test he did not do, Melendez-Diaz violation.
Overruled.
So just give us the general outline of how LIBS works.
Okay, LIBS, laser-induced breakdown spectroscopy, is a laser-based technique. And essentially what happens is a laser beam, in this case, it's a YAG laser. It stands for yttrium aluminum garnet. That's the crystal matrix. So it's a YAG laser. It creates green light, 532 nanometers. So that laser is sorry, I'm sorry.
Slow down.
I know, I
Take your
get
time.
excited when I know something.
That's okay.
Where was I? I'm sorry.
Crystal matrix.
Yeah, the crystal matrix is called a YAG laser, yttrium aluminum garnet. It's a crystal.
I think you've gone into the more than we need to know zone. So let's just,
Well, I like to say the word YAG, so
what does the laser actually do to a
the
substance being tested?
the laser is directed onto the carpet. What happens, elements are composed of atoms. Atoms composed of, the nucleus of an atom is a proton and neutron surrounded by electrons. These atoms can be in two different states, a ground state or an excited state. Okay? Though when the laser beam hits the sample, it creates an excited state. It sets up a plasma, a lot of heat is generated, so the electrons are excited into an excited state. When the laser is shut off, the electrons fall back down to a ground state, an unexcited state. Now, what's cool about that is that as the electrons fall back to the ground state, they emit the energy that they acquired from the laser.
He's testifying outside of his area of expertise, did not conduct this exam.
Overruled.
Go ahead.
Okay, I forget what it was, but as the laser is turned off, the electrons go from the excited state and fall back to the ground state.
Yeah,
Apologize.
that's okay.
Okay. The energy that's produced during excitation of the electrons has to go somewhere as it falls back to the ground state. That energy is released as a photon of light. That photon of light is picked up by a fiber optic and goes into a little spectrophotometer that can detect it. Now, what's interesting is that every element produces a characteristic light signature. So LIBS can be used to identify the elements that are present in a particular sample. That's about as simple as I can make it.
Sure. In this case, was that technique used on the carpet sample?
That is correct.
Were you present when that occurred?
No, I was not.
All right. Did you utilize the results of that evaluation in your eventual opinion and report?
I did.
Now, what is the significance of the results of the LIBS examination of the carpet?
As I believe I mentioned earlier, the human body is made up of both organic and inorganic components. as decomposition progresses, inorganics are liberated. We were looking specifically for elevated inorganic components consistent with a decompositional event. An example is calcium, sodium, potassium, things of that nature. So we were using the libs to non-destructively examine that carpet to determine whether or not these elements were increased in that sample compared to the control sample.
May I approach the witness, Your Honor?
You may.
Exhibit NQ, recognize that as one of the graphs of the output of the libs. That is correct. And is this one of the items you used in coming to your opinion in this case, this graph?
Yes.
Would it be helpful for you to please the jury the results of the test to use this graph?
Yes.
This time you ought to move down to State's Exhibit NQ.
We would object, Judge. This is outside of the witness's area of expertise. Also, I'd like to voir dire the witness as to his methodology as it pertains to this particular item of evidence.
You may proceed. As to that particular item.
It's not in evidence, so.
Dr. Vass, good afternoon, sir.
Hi.
Good morning. You're not a physicist.
Correct.
And Dr. Madavi Martin is the person that conducted this LIBS examination.
That's correct.
And as it relates to Dr. Martin, she's the one in charge of the LIBS laser, correct?
It's her laser, yes.
In fact, you're not even allowed to touch it.
Objection, relevance to the issue.
Sustained.
Methodology, Judge.
Not that question, sustained.
Have you ever utilized this instrument?
Objection, relevance to the issue of the methodology as counsel.
Sustained.
I have used lasers. I'm laser qualified.
But you're not allowed to answer. You have no knowledge of how this test was conducted.
Yes, I know the procedure.
You were not present.
No, but the procedure is well established.
My question is, you were not present.
Correct. I was not present.
In fact, you were not present when she even collected the sample from your office.
Objection,
Were you?
relevance to the issue.
Goes to the methodology, Judge.
Sustained. Let's remove the jury. Chapter 9704 of the Florida Evidence Code provides the following. The facts are the data upon which an expert bases an opinion or inference may be those perceived by or made known to the expert at or before the trial. If the facts or data are of a type reasonably relied upon by an expert in the subject to support the opinion expressed, the facts and the data need not be admissible in evidence. Professor Earhart, in his publication, Earhart on Florida Evidence, the 2010 edition, states the intent of the drafters of the evidence code was to permit expert witnesses to reach their opinions and explain them in almost the same manner as they would in their laboratories or office, citing Swartz versus State 695, 7th 2nd 452, a 1997 4th District Court of Appeals decision. They quote a portion of Swartz, which says, the purpose behind the adoption of this statute as a part of our evidence code was allow experts to use information when rendering opinions in court, just as they would rely on their opinions for examples, nurses, technicians, other physicians, in hospital records when they are rendering opinions out of court. Mr. Baez, I was under the impression that you were wanting to attack some methodology. That wasn't exactly where you were going with your questions. So to be abundantly clear, expert witnesses are able to rely on reports of others in formulating their opinions. The evidence code even goes on to say, even if the evidence may not be admissible, as long as it is something or the type that are reasonably relied upon by the expert in the subject to support their opinions. What you may go into are things that you were going into a proper subject for cross-examination, but not necessarily dealing with what you indicated that you wanted to do. So we have an idea where you're going. You may continue your proffer so we can see the point that you were trying to make. So you may proceed.
Dr. Vass, the results of the LIBS examination, what exactly were they?
They showed that inorganic elements associated with human decomposition was significantly elevated over what was found in the control carpet sample.
And the inorganic chemicals that were found were calcium?
Among others.
Sodium?
Can I look at the report and,
yes, you may.
confirm?
Yes, you may, sir.
Yes, we looked at calcium, magnesium, sodium, carbon, iron, and that was the extent of the ability of that LIBS instrument.
Now, you do not regularly conduct LIBS examinations, do you, sir?
Personally, no.
And these items that you have identified are commonly found in numerous other items in the environment.
Elements are found in everything known to man, yes.
And because this is somewhat out of your expertise, you're not a physicist, right?
Correct, I'm not a physicist.
You can't assign a certain amount of weight as to the importance of these chemical compounds, can you, sir?
I don't know if that's true. I mean, I know which inorganic elements are liberated and are elevated during decompositional events. So I think I can make not logical, but I can make a conclusion that since all these elements, inorganic elements are elevated, that should be elevated, it's just another corroboration of what my nose tells me is correct.
And these are your opinions that would only apply to the variables of which you have studied, correct?
I'm sorry, I'm sure I don't understand that question.
You're testifying that you would be able to gain some knowledge from these chemicals as they relate to the studies that you've conducted, correct?
Yes, so that the inorganic studies showing which inorganic elements were elevated during human decomposition. That's correct.
And those, however, that comparison or that comparative analysis could only be done upon the types of studies that you've conducted.
What do you mean by types of studies? You mean the LIBS studies?
The conditions, the variable conditions of which you have studied. Buried bodies in a body bag, examples like that.
Well, the inorganic work was done on surface decomp.
And my question to you, sir, is that the comparative analysis can only be done of which compared to something you have actually studied.
That's not true. You can compare anything you want. I mean, even if you're just simply seeing what's in one material compared to another material.
So you can compare you can compare the chemical output of a libs to a buried body or a surface body, and it makes no difference.
I don't think you quite understand what's happening here. In this particular test, we were looking at what inorganic elements were present in the spare tire cover carpet material. We compared what was found in the trunk of the car to what was compared in our control samples from the junkyard. That is the comparative analysis we were looking at. You cannot compare a soil sample to a piece of paint chip on a wall. That would not be a fair comparison.
And that is the only comparison that you can make. The controlled sample from a junkyard in Tennessee to the control sample of this car in this case.
Well, that is the one we did.
That's my question to you, sir, is that is the only comparison you can make, and that's the only opinion you can give.
Objection, compound question.
Overrule at this time. He's proffering.
This is my final question, Judge. Can you answer the question, sir?
I'm sorry, can you repeat the question, please?
The only comparison that you did was the comparative analysis between the junkyard sample and the car in this case.
Using libs, that's correct.
And that's the only type of testimony that you can give on this matter.
In terms of libs, that's correct.
I have no further questions, Judge.
I don't have a question for the prosecution.
Any additional objections to the testimony?
We would just object to this witness testifying, A, outside of the scope of his area of expertise, and B, outside of the scope of the comparative analysis which the witness claims to have conducted.
Okay. Objection will be overruled. I'm going to ask both the state and the defense over the noon recess to read page 745 of the 2010 edition of Earhart, the first two full paragraphs, particularly dealing with the foundation requirement that must be laid before experts can regurgitate other things. So we can move a little bit more expeditiously through this matter. Okay, let's return to jury.
Point of moving into evidence states exhibit NQ. I think that's where the objection came up. Can we repeat that from the jury or?
Objection will be noted and objection will be overruled. Let's return to jury. We'll announce that when the jury gets back in. May be seated. Okay, Mr. Ashton, you may continue.
May I publish, Your Honor?
You may publish.
Hey, Doctor, do you have the chart on your screen there?
Yes, sir.
Okay. If you could just explain to the jury what this chart shows.
Essentially, every element comes off at a particular wavelength that's characteristic to that element. In the case of calcium, it comes off in two particular spots. Calcium there, oops, I'm not good at this. Ca means calcium, and this is just one example of the inorganic compounds that we use libs to analyze. The control sample here points to the level of calcium seen in the junkyard carpet sample. And you can see that the bottom line there indicates the level of calcium. It's essentially not even present. These samples here and here indicate the amount of calcium present in the Florida trunk car. Now, calcium is one of the key inorganics we use in decompositional event determinations and postmortem interval determinations. And similarly, the other inorganics are in relatively similar elevated levels compared to the controls.
So we didn't include the charts of all the other elements, but you said they're similar in terms of the...
They're essentially always higher than what the control was in every case. The y-axis, this here, just indicates intensity
All right.
of the response.
Thank you. You can take that down. All right. So we've talked about the GCMS of the odor, of the gas. We've talked about the libs. Did you also perform any kind of analysis, additional analysis of the carpet itself for other compounds?
Yes, we ended up eventually doing some very minor chemical extractions.
And how does that? Did you do that, or was it done in your presence?
I did that.
You did it. What did you do?
Essentially what we did is, of the carpet, what we did is we cut off a few of the fibers and placed it in a solvent. That solvent happened to be methanol. We let it sit overnight in the methanol. The solvent will dissolve things out of the carpet fibers. You then inject a small amount of the methanol into the GC mass spec system and get a chromatogram and identify the peaks, the compounds present in that as well.
Was the purpose of that to detect compounds that weren't going into gaseous form?
That is correct. Okay. Yeah, again, headspace analysis, looking at the gas over a sample, is just what compounds are being evaporated off that sample. So we wanted to delve a little bit further and look at what compounds may be actually stuck into the fibers that would not evaporate very easily.
And what did that analysis find?
Essentially, about the only peak, I mean, there were a number of interesting peaks. I'll have to look through the report to find out.
Slow down a little bit, because I can see the court reporter trying to keep up. So
okay.
Go ahead.
Basically, what we saw was the presence of butyric acid.
What is butyric acid?
Butyric acid is one of the volatile fatty acids that we use for postmortem interval determination. I mentioned earlier it's butyric and valeric and propionic. What's interesting about butyric acid is that is the first compound that's liberated during human decomposition of the fatty acid sequence, or the volatile fatty acid sequence.
And that was actually in the carpet itself.
That is correct.
All right. Did you do any other instrumental examinations of the carpet piece? Significant ones?
I don't think I would call any.
Okay,
we did other tests, but not of the carpet.
Now, I want to go back for a moment and get away from science and get to something more basic. When you first obtained the can that we've talked about with the carpet sample in it, you said it was sealed, correct?
Yes.
When you first opened it, what was your reaction?
I essentially jumped back a foot or two. The odor was extremely overwhelmingly strong. And I was shocked that that little bitty can could have that much odor associated with it.
And did you immediately recognize the odor that was emanating from the piece of carpet in the can?
Yes.
And what did you recognize that odor to be?
It was, I recognized it as human decomposition odor.
That you've smelled many, many times before you've talked
20
about.
years worth.
20 years worth. Now, were you also given another item of scrapings from the wheel well in the car?
Yes.
Okay. let me show you what's been marked the State's Exhibit for Identification JM. And do you recognize the outside of it?
Yes, because my initials are, yes. So I
Your
do.
initials are on it. Okay, very good. And what did that contain?
If I remember right, now, and I'm not, it's been a while, but I think it was a little glass vial containing scrapings from what was from the tire well or whatever it was.
Would you, let's go ahead and open it up
Okay.
and have you take a look and make sure that it's what you recall. Is that what you recall?
Yeah, it ended up being plastic, not glass, but yeah, that's, I do remember now that I see it, yes.
Okay. This time you ought to move to Evidence State's Exhibit JM for identification.
What says the defense?
We would just renew all previous motions and objections, in addition, chain of custody.
Noting previous objections, it would be admitted in evidence as states numbered
125.
Now, did you perform any type of examination on this item?
Yes, we also did a chemical extract of that material.
And what did that reveal?
Again, a variety of kind of ubiquitous compounds, but one of particular interest was acetic acid.
Why is acetic acid of particular interest?
Well, initially, because acetic acid is a byproduct of human decomposition, for one, but also it's a byproduct of manufacturing chloroform.
Now, were you also submitted some paper towels or napkins of some sort for your evaluation?
Correct. I obtained those from Dr. Neil Haskell.
Let me show you what's marked the State's Exhibit HP. Again, tell me if you recognize the outside, and then we'll open it up. And,
I don't remember the box. I mean, it's a box, okay?
let's go ahead and open it up.
Right.
Let me take it over here and do that safer. All right, if you could just take a look at the items without pulling them out in front of the jury, not of evidence.
Yes.
Do you recognize the items contained within State's Exhibit HP as the items that you were sent by Dr. Haskell?
Correct. Yes.
This time you're going to move this to evidence, no additional admissibility, and I'll explain what the next one.
Our previous motions and objections, and also chain of custody.
That's what I'm going to explain next.
approach. Madam Court Reporter. You may continue.
Doctor, when you received the items we were referring to in State's Exhibit HP, in what condition did you get them? Were they in a bag, box?
I remember there being a white bag, a trash bag associated with it, but in terms of the box, I don't recall.
Was it sealed in whatever it was sent to you?
Oh, absolutely.
With tape similar to what we have here?
Yes, I don't remember if it was exactly that, but it was in a box.
But it was fully sealed.
Yes, absolutely.
This time you ought to bring my question.
Inspect the box from a moment before.
Pardon?
May I inspect the box? I may have an additional point to make.
Go ahead.
I would just renew our previous motions and objections. We have nothing additional.
Okay. It will be conditionally received into evidence at this time.
State's Exhibit number 126.
This might be a good time for our daily recess, Judge.
Okay. All right, ladies and gentlemen, the jury is approximately 2 minutes before the hour of noon. We're going to recess for lunch. I'm going to ask you not to discuss this case among yourselves nor with anyone else, along with all of my other previous admonitions. Any additional instructions from the state or the defense? Okay, we'll be in recess until 1:30. The following movies has been asked to be added to the approval list. And I give you copies of these. Blue Streak, A Love Song for Bobby Long, The Proposal, 21, 51st Dates, Dirty Love. Please have your staff evaluate these and let me know by the end of the day if you have any objections to those titles. Okay. Is the witness outside? Okay, let's have the doctor resume the stand, and after he gets back on the stand, we will return the jury. Okay, members of the jury, did you heed my previous admonitions not to discuss this case among yourselves nor with anyone else? Okay, state recognized presence of the jury.
Yes, sir.
Defense? Yes, sir. All right, you may continue, Mr. Ashton.
Thank you. Doctor, let me just get them. I think where we left off, we had just talked about State's Exhibit 126, which are the paper towels, napkins, whatever you want to call them, that you were sent by Dr. Haskell. Did you submit those to some sort of instrumental analysis as well?
Yes.
What type of instrumental analysis did you use on those items?
We also did a chemical extraction in methanol of that material.
Same thing, you told us, take a little bit of it and dissolve it in methanol. That.
Essentially both stained and unstained sections.
And did you run that through the GCMS to see what it would show?
That's correct.
And what were the results of that analysis in State's Exhibit 126?
The reason that was done was because insect pupae and were...
Well, let me stop you because I think you're about to tell us what someone told you, and I don't want you to go there. Just tell us what the instrumental analysis said of the items.
It revealed that there were a number of fatty acids present on the paper towels. These fatty acids included palmitic acid, oleic acid, stearic acid, palmitoleic acid, and myristic acid.
And what is the significance of those particular substances?
Those particular fatty acids are the composition of, make up what is called adipocere or grave wax. It is a byproduct of the breakdown of fat.
Is it associated with decomposition?
Yes.
Now, I believe you also testified about receiving back from the sheriff's office the triple sorbent traps that you identified. I think there's 8 of them from various parts of the garage. Did you analyze those as well?
Yes.
What is the purpose of your requesting those air samples from various areas within the garage or the car?
We wanted to confirm that the carpet sample was the point source of the odor.
Did you also obtain a air sample, triple sorbent trap, from some garbage that had been in, by report, had been in the trunk at some point?
Yes, sir.
And by examining those triple sorbent traps, what did you come to the determination as to whether the carpet was a point source of the compounds rather than anything else?
That is correct. We confirmed that the carpet was the point source of the odor.
And how did you do that? Just tell briefly about what the results were that told you that.
Again, we're looking at a combination of chemical compounds that are present. All the other components we looked at, the car interior, the trash, the garage air, they contained a few compounds of relevance, but the majority of relevant compounds were found in high abundance in the carpet sample.
And were those same group of compounds found, for instance, in the trash?
No.
And how about in the garage air itself, just the garage air from where it was held?
No.
All right, now let's take a step back for a moment and talk about the actual compounds that were found in the cryotrap concentrated sample from the carpet. What is the significance, if any, of the compounds that were found in that sample?
I will answer the question, but can I elaborate?
Sure, please do.
Odor analysis. For example, if you leave here today and you're walking down the street, you will encounter many odors, from flower fragrances, car exhaust, perhaps a restaurant cooking food, perhaps trash. Each of those odors is comprised of a number of chemicals. It could be dozens of chemicals. It's the concentration and combination of those that make each of those fragrances or odors unique. Now, one of the interesting things is that these chemicals are really not unique in nature. So there's going to be cross-reactivity to a small extent. For instance, a rose may have, the fragrance of a rose may have 12 chemicals that make up that fragrance. Okay, and trash, for instance, may have another 20 chemicals that can make up that odor. Now, there's going to be some small interaction between all the chemicals on the rose and the chemicals in the trash, but it's the total combination of chemicals present in each source that make it unique. Now,
let me ask you a question. When you say interaction, do you mean some commonality? In other words, a compound may be in the smell of a rose and in the smell of trash.
Yes, you may have 10 compounds in the rose, and maybe 2 or 3 of those are also found in the trash, but there's going to be another 15 that are different in the trash, giving each separate entity its own odor signature.
All right. Thank you.
Now, in the car sample, the car trunk sample, we identified 51 individual compounds. 41 of those were related to human decomposition based on the studies we've done over the last several years. Of those 41, we had, of course, overlap with gasoline. We break down into gasoline components, like benzene, for instance. Because we didn't know if the gasoline vapors in the trunk of the car originated from a potential decompositional event or from gasoline, we essentially eliminated those to avoid any possibility of confusion. That left approximately 24 compounds. Of those, we then started looking at control samples. We started eliminating compounds that were found also in the car trunk. The control from the junkyard. We looked at compounds from a decomposing pizza. We looked at a variety of squirrel remnants. We looked at the interior of the car components, the garage air, and subtracted those compounds that interacted or correlated with what we saw in the trunk of the car. That left approximately, I believe, 16 compounds. Of those 16 compounds, 7 of those are considered significant, meaning that in the 2008 paper that we published, we made a list of 30 compounds that we considered relevant and significant to human decomposition. And those, of the huge number of compounds we found during human decomposition, which is about 500, we pared that down to 30. And that was based on a number of decision tree parameters, which means we looked at the longevity of the compound, whether the compound's found in human bone or not, whether it's normally found in the environment, how unusual it is, if it's in surface decomposition, what chemical class it falls into, how the environment affects it. So those 500, roughly 400 odd chemicals were pared down to 30 significant ones. In the trunk of the car, we found essentially 13 from that list, but we had to eliminate some because there were gasoline components. That left 7.
Let me stop for just a second and
Is that
make
too
sure
much?
we make this clear. Just because a compound overlapped with gasoline, does that mean that the compound is not there because of decomposition?
No, we don't know where the source is. It could have been from decomposition or it could have been from gasoline. But to be honest and fair, we had to eliminate from the list because we didn't know where the source was.
And because it's in the back of a car and one would expect to see gasoline vapor
Absolutely.
near a car.
Overruled.
Okay. Now, these 30 compounds that you've discussed going through your decision tree of your 2008 paper, are those all compounds that appear at the same stage of decomposition?
No, they are not. It's a study that comprised what we consider the most relevant compounds encompassing burials, surface decomposition, so that differentiated anaerobic and aerobic compounds, as well as, what were some of the other ones? I lost my train
That's
of thought.
okay. Well, let me ask you another question. Maybe we'll come back to you.
Oh, I'm sorry. I can't.
Go ahead.
I'm sorry. Or whether they were early in decomposition or late in decomposition. The interesting thing about human decomposition is it's not steady state. It's cyclic. Decomposition is cyclic. That's because bacteria and microorganisms degrade things at different rates. So you have cycles of decomposition. Early decomposition products are significantly different than products you see late in the decomposition process. So those 30 compounds included aerobic, anaerobic, early and late. So it's highly unlikely that at any one particular time point during the decompositional cycle, you would find all those compounds present.
All right. But the 30 were chosen because they were important at some stage.
That is correct.
All right, so eliminating the gasoline components, eliminating the components that might have been in the control samples, you said you had 7 left.
Yes.
All right, and what was the significance of those 7?
Those 7 were found to be in the list of 30 compounds that we considered most relevant out of the entire 500 odd chemicals in human decomposition.
Now, is there a specific established chemical odor signature for human decomposition? A clear and specific one to human decomposition only?
I do not think so.
So, based upon that evaluation of the odor itself, can you come to any opinion about the source of the odor that you analyzed from the carpet?
I consider it consistent with human decomposition.
Now, you also examined, I believe we talked about the, pardon me, the scrapings from the tire well.
Yes.
You talked about what you found there. Again, does that how does that fit in with your opinion based on the odor that it's consistent with human decomposition?
Again, the acetic acid, acetic acid is a product of human decomposition. It is just not one of the most significant ones that we identified in the list of 30.
Is acetic acid also found in many, many other?
Sure, it's in vinegar, for instance.
I'm sorry, what?
It's in vinegar. Vinegar is acetic acid.
So that's consistent, but not all that definitive.
Correct.
Okay. How about the examination, the LIBS examination of the spare tire cover, the inorganic compounds that were found there? How does that fit into the overall picture of consistency with human decomposition?
Again, all the compounds, the inorganic compounds that one would expect to see elevated in a human decompositional event were indeed elevated.
But again, those are compounds that are found in elements, that are found in other
Correct.
ways. All right. The examination of the carpet, the spare tire cover, the extraction that you told us about. And what did you say you got in the extraction again? The extraction of the spare tire cover carpet.
Yes, butyric acid.
Butyric acid. How does that fit in, again, to the overall picture of the consistency with human decomposition?
Butyric acid, we typically don't see a lot of environmental control samples. That, again, is a product of human decomposition, early decomposition. So it, again, is very consistent with a decompositional event.
You said that you don't commonly see it in environmental samples. Can you explain what you mean by that?
I would say that it's usually metabolized very quickly by microorganisms if it's in small quantities. So you really don't see it in the environment as often. That's about it.
Okay. And lastly, the paper towels that you examined and what you found on those. What is the significance of that in terms of assessing this consistency with human decomposition?
Again, the definition of adiposia, which is the breakdown of adipose tissue or fat, essentially includes every one of the fatty acids that we identified on the paper towels.
Now, is adiposia limited to human beings?
No.
Is there anything about that particular finding that would say it's human as opposed to some other animal or something?
I do not think that adiposia alone can differentiate it. I mean, there are reports in the literature that elevated sodium in adiposia is more human-like than elevated potassium, which is more in pigs, but we didn't delve that deeply into that analysis.
All right. All right. Now, taking all of these instrumental examinations that you have discussed with us today, adding to that your olfactory observation of the carpet itself, the smell, do you have an opinion as to whether the evidence you found in the trunk of this car, let me rephrase that, do you have an opinion as to whether there was a decomposing human body in the trunk of that car at some point?
I do have an opinion on it.
And what is that opinion?
I can find no other plausible explanation other than that to explain all the results we found.
No further questions.
Cross-examination?